To this computer. I Hey Milo, I'm sorry about that. I actually just did. For some reason, I uploaded it and I I must have. Okay, here's what it is. I posted it, but I wasn't ready to post it, so I posted it as a private one, and then I posted it just now, like 20 minutes ago. Sorry about that. Um, the I got a private message of is there a practice exam number 4? Yes, there absolutely is. I think that I included that in my announcement last week, or I thought that I had one. Um, but if you go to quizzes, practice exam number 4, yeah, it's up. It's 25 questions, unlimited attempts. There is a timer on it though, so that you guys can, kind of simulate your exam day type of experience. All right, so today what we are going to do is we are going to be wrapping up our our deal with our deal with enzyme mechanisms. If you remember on Friday, we left off talking about serine proteases. And so we're going to pick up there. But the first thing that we've got is a top hat question. And that is just as a little bit of a reviewer. Which of the following is not one of the three catalytic mechanisms or mechanism types that we investigated and that we talked about? So I think that I've got a timer of about 30 seconds on that one, just because that's a pretty straightforward question. We got 10 seconds left. I bet we can get to 20 answers on this one. In fact, I know we can. Done. 20. Perfect. Great. Acid base, covalent, or metal ion are our answers. We got 23, 24 answers. 25. For some reason, the timer's still, well, 27. Why is it not stopping? Okay, cool. I guess there's a little bit of delay there. The one that we did not investigate was alterado. That's not a real word. I made that one up. We talked about acid-base catalysis, and our example for that was RNase A. Covalent catalysis, we talked about the enzyme lysozyme. And then metal ion catalysis, we talked about the enzyme carbonic anhydrase. Now, just as a little detail regarding each one of those, um, in covalent catalysis, we have a hallmark of covalent catalysis is a covalent bond is formed. You've got a nucleophile and you've got an electrophile. The nucleophile is going to be on your enzyme or one of the amino acids in your enzyme, and then acid base. We have basically a amino acid that's protonated, well, proton transfer. That's effectively all that there is. And then metal ion catalysis. We only have a metal ion, zinc, zinc 2 plus. I'm just going to put an example there, or as an example. Um, Zinc basically gave our enzyme, in that case, carbonic anhydrase, an ability that it otherwise did not have. Now, just as a quick rundown, I'm going to add these RNase A, and then this was lysozyme, and then last but not least, carbonic anhydrase. So those were our enzymes that we looked at for each one of those different answer options. Now, I don't want you to memorize those mechanisms or anything like that. I want you to know the amino acids that participate in those because those are really great models. And I'll ask questions like, what does this amino acid do? And I'll give you a snapshot of it. So if you look at a histidine residue and see that it's protonated, well, chances are that's going to act as an acid. If a histidine residue is deprotonated, well, then it could do a couple of things. It could act as a base, It could establish a metallic bond with a metal ion. It could also work to establish a covalent bond. So histidine is actually kind of a pretty interesting residue because of its kind of, I guess you could say how dynamic it is. Um, okay, so I can clear my drawings there, and now we'll proceed. So we talked a little bit about proteases. Proteases are proteins that cut proteins. They're kind of cannibals in some respects. Which funny story, my 4-year-old the other day said the word cannibal, and that my wife heard this and she was a little bit taken aback by that. And as it turns out, he was trying to say cannonball, like when you jump into a pool. Um, what do proteases do? Well, they work by cutting peptide bonds. They need a dipeptide at very least, and they can't do anything with a single amino acid. So they're looking for a peptide bond. Now, serine proteases are a class of enzymes that what they have in their active site is a serine residue. There are also proteases known as aspartic proteases, and they have an aspartic acid within their active site. Now, it's not just, oh, they happen to have that, but instead that residue is pretty critical for the function of that enzyme. So, serine proteases, some examples, we talked about these a little bit Friday. Chymotrypsin is one, subtilisin is one. And then I mentioned um, TPA, the drug that's administered to people pretty immediately after experiencing stroke-like symptoms. And this is something that has to be administered very quickly. I think it's within the first like 3 hours for its highest effectiveness. And I'm not sure if after that, if it's not used or if it's like less effective. At any rate, this is a drug that's very effective because it's also known as the clot buster. Um, and it basically prevents, a blockage um, from forming, and therefore, part of your brain lose it prevents part of your brain from losing blood flow and oxygen. And I bring this up. My brother a few years ago had a pretty catastrophic stroke, and what was kind of crazy was that he was in the ICU, and I saw another person also in that same ICU who on their door to their room in the ICU, it said TPA patient. And so that person was at a great risk of, if they had a cut, well, they wouldn't be able to clot it. So that would be a problem for them. But the interesting thing was that this person who had TPA administered, they're sitting watching TV eating lunch. Um, their stroke symptoms or their kind of stroke recovery related symptoms were minimal at most. So it's a pretty fantastic drug if it's administered in the right time frame. Okay. So now with that said, proteases, if you think about a protease, it's I'll go back to that term cannibal. It's a protein that likes to eat up other proteins. So if you think about the central dogma of molecular biology, you've got DNA going to an RNA going to a protein. So you have transcription, then translation. Now, if you have a protease as an expressed protein, then it's going to go around and kind of chomp up other proteins. Um, as you can imagine, that's something that you don't necessarily want. You don't want a protease always active, or another phrase there, constitutively active. You want it active under the right circumstances. Now, one way to do that is to go through the central dogma of molecular biology. Like, say that the cell receives a signal, hey, we need to express this protease. Okay, well, cool. Well, then let's go back to the genomic DNA and make this part of the genome active. So then we can transcribe it. Okay, then after we transcribed it, then we can translate that transcript. Well, that's almost like that part of the genome is dormant. It's not doing anything. So you have to activate it. So it's almost like you're waking up that protein in some respects. I mean, not exactly, but you're waking it up. You're trying to rise it so that it's ready. Well, there's going to be a delay there, and that takes time. So as an alternative, rather than going through this process of initiating transcription and subsequently translation, proteases actually are translated as something known as a zymogen. I really love this word just because it sounds like it's from a sci-fi movie from like the 1950s or something like that. But a zymogen is a particular form of a protease. So a zymogen, as is captured right here with this enzyme right here. This is chymotrypsinogen. Now, if we jog back for just a second, if we walk backwards, well, one of the proteases that we mentioned was this right here on the left, chymotrypsin. Okay. Chymotrypsin versus chymotrypsinogen. Okay. Chymotrypsinogen is the protease that is translated. And whenever that protease is translated, it exists as a zymogen or in its zymogen form where it's inactive. Now, what that means is that zymogen basically floats around the cell. It's living its life. It's just like, oh, look at that. I'm not doing anything. I'm not breaking any bonds because there's been no reason for me to break any bonds. So a zymogen is an inactive protease. Now, in order for that protease to become active, what will happen is that protease, as is the case with many other proteases, they undergo what's known as a cleavage by another protease. So here what we have is chymotrypsinogen, the inactive form that is activated by another protease, and this particular protease is known as trypsin. Now, what trypsin will do is it's going to break a peptide bond right there. Okay, so it's going to break a peptide bond between amino acids 15 and 16. And whenever it does that, well, then chymotrypsinogen is converted to pi chymotrypsin. Okay, so that's an active form of an enzyme. That, just by virtue of breaking a single peptide bond, pi chymotrypsin can then go on to break other peptide bonds. It's almost like it's awoken that sleeping giant. So pi chymotrypsin can go on and cut peptide bonds. But what it does first, in order to become the fully active form of chymotrypsin, is it goes through what is known as autocleavage or autocatalytic activity. So auto meaning self. So it's going to cut some of its own peptide bonds. And in particular, what it's going to do is it's going to cut between amino acids number 13 and 14. So right here, 13 and 14. Now, trypsin cut between 15 and 16. Now, my question for you guys in the chat and answer this is a yes or no or thumbs up, thumbs down. Um, is the peptide bond between 14 and 15, amino acids 14 and 15, is that intact? Absolutely. Yes, absolutely. Great job, y'all. So the residues or the peptide bond between 14 and 15 remains intact. And so then when pi chymotrypsin breaks the bond between 13 and 14, what we have released is a dipeptide, the serine and arginine, 14 and 15. What also happens is this pi chymotrypsin cuts between right here, and I'm going to put the numbers above it. So 146 and 147, and then between 148 and 149. Okay, so it's going to basically cut out two dipeptides. Now, a point of clarification, this is not the sequence in which every protease is activated. Different proteases are cleaved and processed in different ways. This is the way that chymotrypsin is cleaved, and it cuts out these two peptides. I don't want you to commit this to memory and say, oh, well, you gotta, in order to activate a protease, you have to cut out two dipeptides. No, that's just this particular case. When those two dipeptides have been removed, however, we've got our fully active form of pi chymotrypsin. Now, what's pretty interesting to me about this and I imagine you guys will think it's kind of neat too, is that pichymotrypsin, it's active, but it's three individual peptides, which is kind of crazy. But those three individual peptides, what residues are responsible for holding them together? Because this right here, uh, circle there, what amino acid is critical for chymotrypsin to remain as three distinct peptides that are held together? I'll give you a hint. This S is your big hint, and a little bit of a spoiler, and to kind of lead into cysteine. Absolutely right, Morgan. So it's a cysteine residue. So these right here, these yellow lines, in fact, all of them, I'm going to put a check mark by some of these here, here, and here. Those three that I just put check marks by aren't responsible for holding individual dipeptides or individual peptides together, but they are also examples of disulfide bonds or disulfide bridges, whichever way you want to split it. So I think that's pretty cool that this peptide, just to give you a little bit of a, physiological relevance or interest. If this residue right here, if this, I think it's amino acid number one, I think it is an N-terminal cysteine. If this were mutated to a serine, for instance, well, if that were mutated to a serine, then the person who had this or the cells that had this mutation, they'd run into some problems. They probably would not go through apoptosis properly or a number of other different cellular processes. They would probably run into problems there because, well, they wouldn't have a functional chymotrypsin. Because if you can imagine, if this bond wasn't here, if this disulfide bond was gone, and a peptide bond were cut between there, and I'm going to label it as, I'm going to do some erasing just so it's a little bit more clear. If you don't have that disulfide bond, but you break the peptide bond that's found between peptides A and B, when you break that bond, what happens to peptide A? Gone. It's gone. It's going to float away. It's going to find some other sort of attraction that it has an affinity for, and it's done. Now, with that in mind, or with that to say, this is the way that many proteases are activated because they're kind of destructive in the sense that they break peptide bonds. Now, they all have specific patterns that they cut after. Um, so what they do is they don't just, they do discriminate and say, well, I'm going to cut between this residue and this residue. But in some instances, that pattern is found quite often in a cell. Now, this is a activation that is irreversible. Um, so as soon as a protease is activated, it's basically set loose. And that can present some problems for a cell. It can, break a bunch of bonds. It can break down important parts of a cell, important processes, metabolic functions, and all that. So that's why I always kind of think about zymogens as like very tightly controlled because you don't want this to be something that just kind of goes without any sort of check or regulation. Okay, now we've got a question, a follow-up question to proteases, and this is going to be a pretty quick one as well. What type of bond is broken to take a zymogen to an active protease? Our options are a peptide bond, a hydrogen bond, a disulfide bond, or an acid bond. And we got the spinning wheel saying to me that it's okay, there we go. 27 seconds left. Oh, all right. We got 35 answers. Wheel's still spinning. What do we got? We can get 36. Okay, I think it's done. All right, let's see the answer. A peptide bond is absolutely correct. So I'm happy that we had some folks answer disulfide bond because that's the type of bond that we need to make sure that basically is not broken. Now, chymotrypsin is just an example, but if you think about a zymogen being activated by a protease, think about what type of bond does a protease break. Now, a hydrogen bond, well, a protease isn't going to break a hydrogen bond. Um, Hydrogen bonds, will a hydrogen bond be broken in the process of activating a zymogen? Sure, sure. But hydrogen bonds are made and broken kind of all the time. And this question was asking what type of bond is like a singular bond. Think about it almost like a one or two or three or something like that. How many of these, um, kind of narrow your scope there a little bit. Okay, next up is when we think about these different proteases, like I said, they have different specificities and selectivities. Now, as an example, trypsin, chymotrypsin, and elastase, these are all serine proteases. Now, this orange shape right here is illustrating kind of a pocket within the protein. So this pocket within the protein, you could kind of think about this as being like the substrate binding site. And what are some residues? What does that substrate binding site have that makes these different serine proteases select for different residues? So, or select for different substrates. On the far left, we've got trypsin. Now, in the this particular sequence or this particular structure is known as a specificity pocket. There we go. It's known as a specificity pocket. And this is basically the um, structure, I'll say, the structure of the protein that selects the right substrate. Now, trypsin is going to cleave on the C-terminal side of basic positively charged amino acids. Now, I want to give you some examples of all three of these. So, first of all, trypsin. If you were to give, remember, trypsin is a protease. If trypsin were to be given a dipeptide that looks like this, RA, okay? Now let's take that sentence. Trypsin cleaves on the C-terminal side of basic positively charged amino acids. Now, I'm guessing that lots of y'all are thinking, okay, positively charged. Okay, what do I know about a positively charged residue? Arginine, lysine, and histidine are all my positively charged residues. Um, but that's kind of pH dependent because if we are talking about a neutral pH, then histidine's probably not going to be positively charged. But arginine and lysine are. And so there is like an element of this that you gotta, look at your pKa's. Now, with that in mind, I'm going to put a little star, and that star on the left-hand side of arginine there, add to the chat, is that the N termini or C termini of arginine? And thinking about the directionality of amino acids and peptides. Absolutely, Morgan. Great. So that is the N termini. Great job, Milo. Thank you. So that's the N termini, meaning that the other side, that's the C termini. Okay. Now, with that in mind, a protease needs a dipeptide. Arginine is a positively charged residue. So I'm going to say R or K. And trypsin is going to cleave on the C termini of this because this is a dipeptide RA. So trypsin will cut here. Okay. Now, follow up. So I'm going to put a check mark that says yes, cuts. Okay. So then if we gave trypsin this peptide, a R. It's a dipeptide. My question for you guys is, will trypsin cut this? And this is kind of a tricky question. Milo, you're absolutely right. This is one that people are always, they're always tempted to say yes. So, Micaiah, you're absolutely right. We need something. So I think, you know, we need something here because proteases are looking for a peptide bond. And if you look at a dipeptide, and I'm going to do some erasing right here. This is our dipeptide. Our N-terminal residue is alanine, followed by that is arginine. What's at the end here? The end is our carboxylic acid. So we don't have a peptide bond to cut. So when you see something like that, it's not going to cut. Does that make sense? Add a thumbs up or a thumbs down if it makes sense, or thumbs down if it does not make sense. A thumbs up if yes, it makes sense. Okay. So I'll do like, one more example here, and then we'll move on to the next one. Okay, thumbs up, thumbs down. All right. So here what we have is we're going to go back to trypsin. Um, and I always like to, whenever I'm writing a peptide, one thing that I try to do is I'll put my N termini. Okay, I'm going to underline that to indicate that I'm not talking about asparagine, but instead I'm talking about my N termini. If we had a dipeptide that looked like this, K A, let's say KALEKL. Now, this is obviously not a dipeptide. Instead, we've got 6 amino acids, so this would be a hexapeptide. Now, what I'm going to do is I'm going to switch colors to green. And first of all, would everyone agree that this is a peptide that trypsin can cut? Awesome. Awesome. Okay. Now the question is, where is trypsin going to cut it? Well, trypsin is going to look for arginine or a lysine. And I always like to narrow it down. Yeah, it says positively charged amino acids, but chances are we're not talking about a histidine. There might be some circumstances at a very low pH, but typically, if we're talking about a neutral pH, this is what we're going after. Now, what is trypsin going to do? Think about trypsin as, I always think about it like a box or something. It's an enzyme. And what it's going to do is it's going to scan this peptide and it's going to say, okay, cool, here's a lysine. I cut after a lysine. So what I'm going to do is I'm going to cut here. And then I'm going to go on to the remaining portion of this peptide. Alanine, trypsin doesn't care. Leucine, trypsin doesn't care. Ooh, glutamic acid, E. I mean, that's kind of interesting because it's charged, but again, trypsin only cares about positively charged residues. So trypsin's going to go here, nothing, don't care, nothing, don't care, nothing, don't care. Oh, but here's another lysine. Oh, now I'm interested. And I'm going to cut on the C termini of that. So if we gave trypsin this peptide, what trypsin would do is it would break it into a couple of different pieces. It would give it a free, lone, um, individual amino acid lysine. So that would be one peptide, and I'm going to put the one over that. And then it would make another peptide, which would be ALEK, and then a third peptide, which is actually just the free amino acid leucine. So trypsin is, as an example, is looking for that positively charged residue. Now, chymotrypsin chymotrypsin and elastase, they all do kind of the same thing. They look for one specific residue and they cut after that, or one specific grouping of residues. And ultimately, this all boils down to what is in the specificity pocket. If we go back to trypsin, trypsin structurally. Great, Morgan. Awesome. That's great. So trypsin's going to have within its specificity pocket an aspartate. Okay. Think about what aspartic acid is charged with, or what its charges at a neutral pH. Well, chances are this residue is negatively charged. And that negatively charged residue is going to attract those positively charged residues. So it's almost like this aspartate, if it sees a phenylalanine, it's like, okay, well, I'm not interested in establishing a connection with you. I like an ionic interaction. If we then look at chymotrypsin, well, the specificity pocket makeup is a little bit different. Rather than having an aspartic acid residue, all of the amino acids that make up the specificity pocket for chymotrypsin are pretty much nonpolar. So it's a bunch of hydrophobic residues. And as a result, chymotrypsin is going to be a peptide or a protease that attracts and cuts after aromatic residues. So if we think about an aromatic residue, we're thinking about, well, phenylalanine, tyrosine, and maybe a little bit of tryptophan there. Um, these are all residues that kind of they're compatible with or they match up with that specificity pocket. Elastase, last but not least, it's going to cut on the C-terminal side of a small neutral residue. So we're looking at like glycine or alanine, maybe a valine, um, just your smaller residues. Isoleucine and leucine, those are going to be good targets to cut after with an enzyme like elastase. Now, a great question related to this stuff is, well, what should I expect in terms of a question on an exam? Let me show you this right here. This is the sort of question that I dig on, where I just say, okay, cool. Well, this is our this is our protease, and what is it going to cut after? And what are the products of that particular um, so this is our protease. Here's a peptide that's being fed to it. What are the products? So those are kind of the big things to me. Okay, cool. Let's proceed from there. Now, clear all my drawings. Here's where we get to the serine protease, the serine part of the serine protease. Now, this serine protease mechanism is a little bit complicated. And so we're going to talk about kind of the important thing to me, and that is what are the individual amino acids doing? In the PowerPoint that I believe that is posted on your Canvas page, I've got this mechanism broken down a little bit more in terms of like, here's all the individual steps, or maybe here's two steps on the process. Um, And that's not to say, well, this is super emphasized. I need you to know this, but instead, that's just if you want to look at it with more detail, here's what you got. So we're going to look at the mechanism for the serine proteases. Now, this is effectively, think about these amino acids as being the amino acids that follow our specificity pocket. So the specificity pocket is what selects the substrate. And then that specificity pocket, or your substrate binding site, is right next to your active site. These amino acids, known as the catalytic triad, are the amino acids that constitute the active site residues. So basically, every single serine protease is going to have histidine, aspartate, and a serine residue. So the active sites of trypsin, chymotrypsin, and elastase, all serine proteases, are composed of this catalytic triad. Now, are there any instances where there's some variation on this? Yes, but they're rare And those mutations or those differences are very, very minor differences. Um, in the event that you had a mutation of one of these residues to a different residue altogether, like in terms of its role, or sorry, in terms of its makeup, well, then that enzyme's probably not going to function. Now, what I do want you to know, there's no doubt in my mind about this. I do want you to know what each amino acid is doing. Serine is acting as a nucleophile. Histidine is acting as a general base. And then aspartate is doing hydrogen bonding, or it's participating in hydrogen bonding to properly orient the histidine. Now, if you've ever seen the movie The Big Lebowski, I always think about this as basically like the Lebowski's rug. Uh, Aspartate is the residue that really ties the room together. If you've never seen that movie, then okay, we're moving forward. Um, so here's our residue: serine, histidine, aspartate. Here's our substrate right up here. Our substrate is a peptide because remember, we're talking about a protease. Now, whenever we look at a peptide, it's always important to kind of identify the 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 bond that is actually being broken. Now, one thing that I like about this is this, okay, so N, C, C, N. Okay, so where's our peptide bond? It's right here. So that's the bond that we're looking to break. Now, with that in mind, if we use this slide, yeah, we're told that it's chymotrypsin, and then we can jump back and look at the specificity pocket if we wanted to, and we'd see, oh yeah, the specificity for pocket for chymotrypsin accommodates an amino acid like that right there, our phenylalanine. Okay, now, like I said, we're trying to break a peptide bond, and this is that bond that we're trying to break. How are we going to do that? Well, if we look at our different amino acids, aspartate, histidine, and serine, well, the first thing that's going to happen, and it's going to happen kind of simultaneously, is that this histidine residue, which is acting as a general base, is going to abstract this proton from serine, which is going to allow serine to establish what type of bond is this? Add to the chat the type of bond that this is. And I'm thinking kind of generally um, between our nucleophile and our substrate, what type of bond do we need to make? Or what type of mechanism needs to have a nucleophile in the enzyme? Boom! Absolutely. I see that you guys' keyboards have that same function that sometimes mine has, where it has a question mark instead of an exclamation mark. Because Micaiah and Darlene, I knew that you guys actually meant to say covalent, and you're absolutely right. It's a covalent bond. Our serine is acting as a nucleophile, and we're establishing this covalent bond. So now the great thing is that we've got a strong connection between our substrate and our enzyme. What that's also going to do is it is going to have, and we're going from 1 to 2. What that's going to do is basically allow kind of our peptide to begin breaking down. Because this carbon right here, which I'm putting a little blue dot on, that now has 4 bonds. And what does that also do? Well, that carbon with 4 bonds, it now has a nitrogen as the second part of that peptide bond. And that nitrogen now it's going to extract this proton right here, and it's going to have a positive charge. And what do we know about positively charged peptide bonds? What we know is they don't really exist because they're unstable. So what we'll have is basically here, our peptide bond is effectively broken. And so the C-terminal side of our peptide, gone. It's with the birds now. Okay. We still have the N-termini of our peptide, whether it's a dipeptide or a protein with 175 amino acids in this R group. That's now covalently bound to our enzyme. Now, what we need to do is get that peptide bond or that peptide to be released. We need that substrate to be released so that our enzyme can go back to doing this over and over again and processing other proteins. In order to do that, we've got to bring in water. Because when we establish this peptide bond and the hydrogen is taken from histidine 57 by the C terminal side of our peptide, well, arguably all of these different things right here are kind of good to go. Like serine with a covalent bond to this carbon right here, it's not perfect, but it can live like that. Histidine, well, histidine's deprotonated, and that's kind of how it wanted to be from the jump. Aspartate, oh man, I always like to inventory aspartate and say, okay, in cell one, deprotonated. Cell two, deprotonated. Cell three, deprotonated. Cell four, deprotonated. And we're going to go ahead and ride this throughout. Cell five, deprotonated. Six, deprotonated. Seven, deprotonated. That aspartate is deprotonated throughout. Why? Well, because it's participating in hydrogen bonding. It's not doing nothing, but it's kind of doing all the behind the scenes work, all the essential work to make sure that the rest of the system doesn't collapse. But this histidine and this serine, I mean, the only thing that's a little bit off is that serine has a covalent bond. Not ideal, but it can work with it. We bring this water in. Now, all of a sudden, histidine again goes back to working as a general base. And whenever it does, it picks up that proton from water. And then we get, oh, lo and behold, water with those lone pairs of electrons goes on to that carbon that has those four bonds. Now, two of them are in the form of a double bond, and then the other two are single bonds, single carbon bonds and a single carbon oxygen bond. When this OH rolls in, what that's going to do is going to push these electrons up towards our oxygen right here, giving that a negative charge, but that's unstable. So then these electrons are going to swing back down, reestablishing our double bond right here, and then causing these electrons to swing over and pick up that proton from our protonated histidine residue. And that will return histidine to being deprotonated, like it was in cell one. And then serine, not in its strong nucleophile form, but in its form where it can go ahead and establish another covalent bond. Now, what details do I want you to know about this? I want you to know the type of bond that's established between serine and the substrate. I want you to know the different states that they're in, the different protonation states, what they could be doing. For instance, when histidine is deprotonated, it's going to work as a good general base. When it's protonated, good general acid. Then I want you to know the roles that they play. Aspartate involved in hydrogen bonding. Histidine involved in, well, proton exchange. It's either acting as a base, which is how it starts and how it's presented itself as, or it's working as an acid, which is what we see later on in cells 2 and cell 6. Serine, it's your nucleophile. That nucleophile is not going to work in a vacuum. That nucleophile is going to work whenever there's that protonated histidine next to it. So I want you to know those things, like know the players, because to me, that's one of the more important things. Pushing the electrons around, yeah, sure, that's valuable, but what we're trying to do is we're trying to lay the groundwork for a firm foundation in what do the different amino acids do and what can they do? And then one question that I think is kind of a next level question that I really like is if you were to mutate that serine to a different residue, which would be a good residue to take the place of that serine? Okay. Leucine. No, that's not a good one. So what amino acids could effectively act as a nucleophile? Well, maybe a cysteine, maybe a threonine, but threonine's kind of bulky. But if we were looking at it and saying what's most similar, well, the most similar residue to cysteine or to serine would probably be cysteine because serine and, or sorry, sulfur and oxygen are similar in their electronegativities, but they are a little bit different in terms of how large they are. But the important takeaways for this slide are what do the different residues do? And actually, that's not this slide, that's the previous slide. What do they do? Nucleophile, general base, hydrogen bonding. And then just if you're thinking, I need to be able to think about this in terms of a movie from 1998, well, remember that the aspartate really ties the room together. because that's the Big Lebowski reference. Came out in 98, I think. My brother was a senior in high school. Okay, so now what we've got, enzyme regulation. So enzyme regulation, it is, well, it's not trivial, but enzymes can be regulated in a couple of different ways. And one way is in which they can be regulated is by different isozymes. So isozymes are enzymes that have similar but not identical amino acid sequences. An example of an isozyme or of a pair of isozymes are hexokinase and glucokinase. Now, what's the difference between these two enzymes? Well, they do the same reaction, but they differ in their kinetics, Km and Vmax. For example, hexokinase is basically in every tissue. Glucokinase is pretty much exclusive to the liver. Now, what makes them different from one another is their kinetic parameters, their Km and their Vmaxes. I thought I put those Kms on there. Now, with that in mind, well, glucokinase has low affinity for, I'll say, hexoses in general, but a high affinity for glucose. And I'm going to put in parentheses just so we can keep this straight. This would be a high Km. And then here, low Km. Because glucokinase loves to bind hexose. Or sorry, glucokinase loves to bind glucose. Other hexoses, not much of an interest. Whereas hexokinase kind of has like an equal affinity for basically every hexose, whether it's glucose or any other glucose or any other hexose. Um, so isozymes differ because, well, they're going to carry out the same reaction, but they kind of have different selectivities. In some instances, it's a tissue difference between them. So that's one way in which an enzyme can be regulated by being only expressed in certain tissues. Another way that an enzyme can be regulated is through a process known as allosteric regulation, which we know a little bit about allostery. We have hemoglobin as our allosteric protein that we've talked about. Now, in terms of what that looks like in a metabolic pathway, I think this is a really good example because what we have is the process where we're taking threonine to leucine. Okay, so that's our start point and our end point. There's a series of steps between those two. Now, one critical enzyme in this whole process is known as threonine dehydratase. That's our first enzyme. That's going to be an oxidoreductase that uses threonine as a substrate. Now, isoleucine, you can think about that as our endpoint. Isoleucine is actually an inhibitor of threonine dehydratase. So essentially, it's almost like an assembly line or a conveyor belt. And isoleucine is the molecule that signals back as like a negative feedback loop, like, hey, stop, stop. We don't need any more of me. I've reached a very high concentration. We're good. Let's stop right there. Now, what does that look like in terms of like structure? So, in terms of the structure of an allosteric enzyme, you have your substrate and you have a positive or a negative modulator. Here we're highlighting a positive modulator. Now, here is your T state of your protein. Okay, so it's not bound to the substrate or the positive modulator or the allosteric regulator. Now, what we also have is, well, that positive modulator binds. And whenever that positive modulator binds, it changes the structure of the protein. It causes it to change shape. And whenever that protein changes shape, then the natural substrate can bind with greater ease. And that can then go on to produce our product. So whenever that positive modulator releases, well, then you go back to that T state, or whenever that positive modulator binds, you move to that R state. Now, I want to get to these last two things, and these are important items. Covalent modification. Think about these as these are going to be good primers for top hat questions for next time. Covalent modification. This is reversible changes to amino acid side chains. These are what are known as post-translational modifications. Enzymes are used to convert the regulatory enzyme to either an active or an inactive form of your protein. There is one modification known as an adenylation, and then another known as a methylation. These are two examples. And what these are is these are molecules that are bound to or added to your enzyme to turn it on or turn it off. And this is the slide that we're going to start with on Wednesday. And then on Wednesday, we're going to talk about lipids. This is important, so that's why I want to give it time at the beginning of our next class period. Um, at any rate, I'm going to go ahead and stop my share. I'm going to say, I hope you guys have a wonderful, what is it? Monday. I hope you have a great Monday, a great Tuesday, and I'll see you Wednesday morning. Deal, everyone? Thank you very much for coming to class. Thank you, guys. Bye.